Hello again. Welcome back to day 2 of the infected cell. So we are going to pick up right where we left off. So when we last met, we were talking about how viruses affect signal transduction pathways and how viruses affect gene expression in their host cells. So today we're going to cover the second half of this topic. So we're going to talk about metabolism. How do viruses affect metabolism? And how do viruses remodel cellular organelles? So we're going to start with metabolism. This image probably brings you back to your 185 days. And so specifically, this image is showing us aerobic respiration, and it's showing us the 4 stages. So glycolysis, formation of acetyl-CoA, the citric acid cycle, and then the electron transport chain and chemiosmosis. So we're going to start with thinking about how this applies to a cell that is infected by a virus. So the first thing I'm going to have you all do is brainstorm. Why do you think viruses manipulate metabolism? All right, so hopefully you've taken some time. And as always, if the things that I am writing don't match the things that you are writing, or if I forget something, please leave it in the comments so that we can have a discussion as a class. So number 1, the main reason why viruses have to manipulate metabolism is they need energy, right? Building virions is no easy process. There's a lot of steps that have to occur. So whenever you are making a virus, this is a high demand on host biosynthetic systems. So what does that mean? Well, if you think about it, our host is not usually making, anywhere from 100 to 1,000 or so virions, depending on the virus. That's a lot for one host cell to make, and you need a lot of energy. So let me give you an example of energy needed. It takes 4 ATP to make 1 peptide bond. That's a lot. 4 molecules of ATP to make 1 peptide bond. And think about the average size of a viral protein. That's a lot of amino acids strung together. And if you think back to aerobic respiration, we only get 38 maximally from aerobic respiration from 1 molecule of glucose. So that's a lot of molecules of glucose that a cell has to break down, a lot of energy that is needed. And this is just for peptides. And of course, we are using a lot of both catabolic and anabolic pathways. So catabolic pathways is going to be breaking down. So breaking down things like glucose, like in the case of glycolysis, and obtaining the energy stored in those chemical bonds. And then anabolic is going to be building. And so of course, we're going to be building a lot of things, right? We're building nucleic acids, we're building proteins, we're building new fats for those envelopes for the viruses that are enveloped. So we're going to be making a lot of things as a host cell. So as a viruses, just like they have ways to manipulate gene expression and ways to manipulate signal transduction, they're going to manipulate metabolism as well. Now that we've talked a little bit about why viruses manipulate metabolism, let's talk a little bit about how and what metabolic pathways they manipulate. So, as a key point, viruses are able to manipulate all of our metabolic pathways. So any of the metabolic pathways that you have learned about, whether it's in Bio 185 or in your biochemistry classes, all of those pathways can be manipulated and changed by viruses. So kind of some of the more common ones that we see at least increases in is we see increases in glycolysis. We see increases in fatty acid production, particularly for those viruses that require an envelope. And there was this really neat experiment that scientists did where over time they measured the amount of glucose that was used. So we're going to do time after infection on the x-axis, and on the y-axis we're going to do glucose usage. And we're going to have the filled in bars be a virus infected cell, and then the empty bars be a mock infected cell. So upon initial infection, both cells had very similar glucose usage. As infection increased, or time after infection increased, I should say, there was an increase in glucose usage and an increase in glycolysis. And this was completed with adenovirus. And although this experiment was completed with adenovirus, this of course is true for many viruses. And so from this experiment, scientists concluded that glycolysis increases in infected cells. And on the previous page, so I'm going to back up 2 pages actually. So on the previous 2 pages, on page 3, I showed you this picture that should hopefully again remind you of Bio334. So there are viruses that increase stage 1. So they will increase glycolysis, and stage 2 at the end, we get production of acetyl-CoA. Well, it turns out that acetyl-CoA can be used to make fatty acids. So some viruses will therefore increase the production of acetyl-CoA, but they will stop the citric acid cycle so that they can siphon off that acetyl-CoA to be made into fatty acids. So let's go ahead and jot that down as well. So some viruses can stop the citric acid cycle, and then they can use the acetyl-CoA to make fatty acids. So the other thing that can happen, although the details are not shown here in glycolysis, one of the things that happens in glycolysis is we make a glucose 6-phosphate. So rather than continuing with glycolysis, we can actually take glucose 6-phosphate, and then that can be shunted off to make nucleotides. So again, the goal here is not to have you memorize all of the ways that viruses manipulate metabolism, but to at least highlight the key ways. So we're going to see energy that is being taken from the host. We're going to see different metabolic processes that are both upregulated and downregulated. Some of them are shunted off to make nucleotides. Some things are siphoned off to make fatty acids. And again, which one is off, which one is on, will fully depend on the virus. Even within the same families of viruses, we can see differences. So the goal here is to just show you the many different ways that viruses can affect metabolism. So with all of these changes in metabolism, just like we have changes in gene expression and changes in cell signaling, there are, of course, repercussions that can happen with respect to the cell. So I'm going to go ahead and have you all brainstorm and think about what kinds of human health effects could this have. All right, so hopefully you have taken some time and you have come up with some thoughts and some ideas of what kind of potential human health effects could this idea of shunting metabolism or turning things on more, turning things off, what kind of effects could this potentially have for human health? So since you are not here in person with me and I cannot hear what you're saying, I'm going to jot down some of the things that students last year shared with me and then have you share any of your other additional ideas in the comments. So some things that we can have is students suggested that maybe there's an increase in fatigue. Absolutely, right? If your cells are on full drive and they are increasing the amount of glucose they're bringing in. They're increasing glycolysis. Yes, they're producing a lot of energy, but that energy is being shunted to make virus. You're probably going to be pretty tired. Students also suggested that maybe there's an increase in heart rate, so kind of more big picture, holistic, whole body things that can occur. Maybe we have an increase in body temp and an increase in inflammation. And all of those things happen. The increase in body temp and increase in inflammation is definitely a way for our immune system to let our body know that there is something wrong. So those are kind of more further down the line things that occur, but absolutely that's something that can happen. The two things that we, however, are going to talk about are more specific, and we are going to talk about diabetes and fatty liver disease. So probably not something that you would have guessed, but just like viruses can cause cancer, viruses can have other health effects as well. And specifically the two things that we are going to talk about is we are going to discuss hepatitis B. So hepatitis B is in the Hepanavirinae family, which is that wonky, gapped, double-stranded DNA. And we're going to talk about hepatitis C, which is a flavivirus. So it's in the Flaviviridae, and it has positive RNA. So both of these viruses infect liver cells, and they cause inflammation of the liver. So let's talk a little bit about the liver and why it's important and why do we get issues with these viruses if they infect. So I'm going to draw, this is my version of a liver. It's my husband's favorite organ. So the liver is important for a lot of things. It is important for glucose homeostasis. We can also store sugar here. It has a key multitude of other enzymes that it produces. But in the case of our discussion, we're going to add that if you have inflammation of the liver, it can affect homeostasis of glucose. So kind of bringing this back full circle now. Why are we talking about the liver, and what does this have to do with hepatitis B and hepatitis C and human health effects? So when we have hepatitis B and hepatitis C patients, one of the things that we see is we see an increase in type 2 diabetes and fatty liver disease. So you might be asking yourselves, why do we see an increase in type 2 diabetes and an increase in fatty liver disease? And the short answer is it has to do with how these viruses affect homeostasis of glucose and how they affect the production of fatty acids. So we just said that viruses can affect glycolysis, they can affect fatty acid production, nucleotide production, any and all metabolic pathways in the host can be affected. So let's go ahead and start with a blank page, and we're going to draw how this happens. So let's start with a virus. So we are going to begin with a virus infection. When that virus infects the host, it's going to increase glycolysis. And of course, the increase in glycolysis is definitely from the viral perspective, it's a benefit to them, so it's definitely a selfish thing. And so when we increase glycolysis in the cells that are infected, they're increasing the amount of sugar that they're bringing in. So now what happens is now we need more glucose. right? We've brought in all of that glucose into the cell, so now we need more. So now we're going to make more glucose. All right, well, now that we have made more glucose, well, now we have an issue because this throws off the homeostasis, which is going to do a couple of things. This is where inflammation comes in. But in addition to that, now we have an increase in blood glucose levels. And short-term, an increase in blood glucose levels isn't the worst thing in the world, right? Our body has ways of dealing with that and then going back to homeostasis. But if this stays elevated for a long time, one of the things that we can see, or that we see in patients, is that type 2 diabetes can develop. So again, seems like a very simple thing. A virus infects, goes through the entire reproductive cycle that we've been talking about. So it increases glycolysis for a little bit. No big deal, right? Until you have hepatitis B, hepatitis C, which have very long infections. So in the case of hepatitis B, which is our Hepanoviridae, although we haven't spent a lot of time talking about the Hepanoviridae family, in addition to the retroviruses, they are the other family of viruses that has reverse transcriptase, which means that they integrate their genome. And so we're talking about latent infections here. So we're talking about a long period of time. And so when these blood glucose levels are elevated for a long period of time, this is why we see type 2 diabetes and many patients that are infected by these viruses. And the same type of process or logic can be walked through when it comes to fats or fatty liver disease. And so instead of glycolysis here, we would change glycolysis out for fatty acid production. So we increase fatty acid production. Now we need more fats. And so what's going to happen? You're going to ingest more of those. More fats are going to come in. It's going to throw off homeostasis. It's going to cause inflammation. And now we are increasing the amount of fats that we have, and we get fatty acid liver disease. And again, these take years to occur, but these are still human health effects that are directly because of viruses. And so these are associations due to viral infections. So hopefully I've convinced you that these viruses can have huge metabolic effects on our cells. So the very last thing that we're going to talk about for this topic is we're going to talk about remodeling of cellular organelles. So we're going to start this part just like we did the other parts, and that is with a discussion question. So knowing what you know thus far from this class, see if you can come up with why might cellular organelles be remodeled and which organelles are remodeled. All right, so welcome back. Hopefully you've had a fun time brainstorming. So let's start with the why part. Why might these cellular organelles be remodeled? If you think back to replication, especially when we talked about Picornaviridae replication, we talked about how these viruses replicate on membrane vesicles. And so many viruses use membrane vesicles for replication. Those membrane vesicles, they're membranes. They come from those membrane-bound organelles. Oftentimes, viruses set up viral factories. So those membrane vesicles are oftentimes converted into viral factories, which are just a site in a cell where a lot of replication and assembly occurs. We think it makes it easier for the virus so that you're not shuffling things back and forth throughout the cell. And so again, we've got these replication sites. And of course, for the viruses that require an envelope, we need to use the cellular organelles to make an envelope. And we can actually start to answer this question of which organelles. So we have seen the membrane for enveloped viruses come from many different places. So the nucleus might be affected. We've seen the Golgi, the ER. So a lot of those membrane-bound organelles that were the ones that we talked about in assembly and exit are going to be the same ones that are going to be affected and remodeled. And I'm going to go ahead and add the plasma membrane onto our list as well. So we're going to focus on two main pieces or two main parts of the cell. We're going to focus on the nucleus and then the cytoplasm. So the cytoplasm contains the Golgi and the ER, so we'll kind of group those together. So let's start with the nucleus. And what happens depends on the virus, right? Most of our RNA viruses stay away from the nucleus, so there's not going to be too much of a remodeling that occurs in the nucleus. We might be affecting, if we're making mRNA, we might be affecting whether we have more heterochromatin versus euchromatin, but in terms of remodeling the nucleus, they pretty much leave it alone. But if you are a DNA virus, herpes virus, adenovirus, these viruses go straight to the nucleus. And so one of the things that happens is the nucleus is going to get bigger. So as that nucleus gets bigger, you're probably wondering, why is it getting bigger? Well, let's go ahead and draw a cell. So here's our fried egg looking cell. The yolk part of the cell, of course, is our nucleus. So for adenovirus, for herpes virus, these viruses assemble in the nucleus. So depending on the virus, of course, these numbers range for viral progeny, but they can be between 50 to 100 to 1,000, depends completely on the virus. But now imagine you have all of our host chromosomes in the nucleus, but now you have hundreds of viruses that are assembling in the nucleus. That nucleus is going to get larger because it has to accommodate those viruses. So I'm actually going to show you a couple of images of what this looks like. So let's go ahead and take a look at a polyoma virus. So go ahead and write this down so when you look back at this, you remember what it is. So for the polyoma virus, on the left, we have early infection. On the right, we have late infection. The CYT stands for the cytoplasm, and the CHR stands for chromatin. So early on in infection, here where this black arrow is, this is the production of the major capsid protein. So let me pick a bright color here so I can write this. Major capsid protein. Here down where we have this white arrow, these are viral particles. So we can see that early on in infection, we have the chromatin kind of up against here, against that nuclear membrane. We've got a little bit of the chromatin here, and we can start to see in spots of the nucleus that we have these viral factories that are being set up. Now let's take a look on the right side. On the right side, our chromatin, hopefully you can see around the edges here and kind of here on the inside, it has gotten much darker. That chromatin has been pushed up and condensed, and we have all of our viral particles, our progeny, that are right here. And so again, this nucleus has gotten larger, that chromatin has gotten smushed into a smaller area to accommodate these viruses. So let me show you another really cool image. And the second one that we're going to look at is varicella zoster, which is a herpes virus. So same virus, two different diseases. So the virus that causes chickenpox and shingles. So on the left, again, we have early infection. And on the right, we have late infection. So here we're looking at a tomogram, so a 3D image of what's going on. So let's go ahead and kind of walk through what's happening. The blue here is the chromatin. So we can't see the whole nucleus, but around the green blobs, which we'll talk about those in a second, that is supposed to represent the size of the nucleus. So that chromatin is being pushed up against that double membrane, so that nuclear membrane. The green that we have, that PML, the PML is actually, so the virus kind of takes over it, but it is a nuclear body, and this is something that is normally found in our host, and our nucleus is made up of these different types of nuclear bodies. There are many kinds that exist, and they're subcompartments, if you will. They're non-membranous subcompartments, though, and many help with genome maintenance repair. Many also help with things like epigenetics, and so control of gene expression on top of the typical things that we would think of. And so what these viruses do is they kind of take over these nuclear bodies and they set up shop. So that's where their viral factories are. So on the left, we can see that we have some capsids that are starting to form, and both on this part of the nucleus and in this part of the nucleus, we see that the capsids are very much so localized to those PML nuclear bodies. Now, if we take a look at the right of what's happening, our chromatin has been pushed even further, and again you can see that it's been condensed, and now we have one giant PML. This nucleus is much larger, and we can actually start to see two types of capsids. So the yellow IM ones, let me make sure I match my colors here. So the yellow IM ones are the immature capsid. So if you think back to our last topic, we talked about how herpes virus actually makes a procapsid, and then that procapsid then expands as the DNA is pumped into it. So it's a head full and genome length packager, and then we end up with a mature capsid. So in this work, the scientists were able to capture both the immature and the mature capsids that are being made inside of this PML, inside of this viral factory. So again, hopefully I'm convincing you that there are significant changes that occur. So the next thing that we're going to talk about is the cytoplasm. So in the cytoplasm, oftentimes membranes are repurposed. So typically when we draw a cell, we draw our nucleus, and sometimes we're good and we draw the second membrane of the nucleus. We draw our ER that extends from the second membrane of the nucleus. Then we have our Golgi. And again, I'm leaving out most of the organelles. I'm kind of focusing on the major ones that are repurposed. But when we have a virus that infects, oftentimes the membranes that would typically make up the ER or the Golgi are repurposed, and they're repurposed to set up viral factories. They're repurposed for autophagy. So again, we talked about how that is a mechanism of exiting the cell. And so again, viruses are doing this as a mechanism to help themselves out. So let's look at some pictures of how this happens. So we're going to look at dengue. So dengue virus is a flaviviridae virus. And we're going to start on the left, which is replication, and then kind of work our way over to the right. So I'm going to go ahead and I'm actually going to zoom in on this picture here. And this is from our textbook, as are the other images. So here is our ER. You'll notice that we're starting to get kind of this weird blob here. This CM stands for convoluted membrane. You gotta love when scientists name things ridiculous ways. So we've got a convoluted membrane. We've got some viral proteases in there. We can start to see that we have the viral polymerase. We have double-stranded RNA. And if we zoom in to see what is actually happening at the molecular level, looking at electron micrographs, we can see the ER is here And here is that convoluted membrane. So this used to be part of the ER, and we can start to see that it's taking on different shapes. And we can also start to see here where these little arrows are pointing. It kind of looks like a pea pod. So if you were to open it up and you see the little tiny peas in it. So this little pea pod here, this is a 2D image. And here is a tomogram shown of that. So the red here is a capsid, and each of these little peas in the pea pod are actually little vesicle packs. And of course, this is all what used to be the ER and the Golgi. So now let's go ahead and look at assembly and what's going on here. So we have our ER down here. This ER now, if we start to look on that, in that convoluted membrane that we had, we're starting to see nice assembled little virions. So for the flaviviridae viruses, they tend to come in, replicate, and assemble on vesicles very closely. So in the ER and the Golgi. And we're going to get these viruses that are then going to go into the Golgi. So if we zoom in here on this electron micrograph, we can actually see a little viral particle. Here is another image where we see a bunch of viral particles. And on the right, we can see that what used to be that pea pod, now we have a ton of viral particles on the inside. So again, this group did a really nice job of showing us how the Golgi and how the ER are completely remodeled and repurposed for the virus. So last but not least, from the virus's perspective, what is the goal in doing all of these things? Why do we affect gene expression? Why do we affect proteins or signal transduction? Why do we affect remodeling of cellular organelles? And why do we affect metabolism? So hopefully you are yelling at the screen and you are telling me that the goal here is to ensure maximal viral production. And of course, when we do this, not only do we want to ensure maximal viral production, but we want to get out before the host immune system responds. That is kind of key and clutch. So what we are going to do next time is we're going to actually skip ahead to viral pathogenesis. So we are skipping Unit 3 and we're going to talk about Unit 4. Unit 4 is going to definitely help us a lot more with the current events in the world. And then after we finish up Unit 4, we are going to circle back around and come back to Unit 3, but we're going to spend an abbreviated time on it and kind of highlight the main points again, those points that are going to help us best understand the coronavirus pandemic that is happening right now. So thanks for hanging out with me for another online lecture. Please let me know if you have any questions, comments, or concerns. You have many ways of reaching me. And until next time, bye everyone.